Hi everyone, Matthew Wood here on Journeys to the Ice, and this is the third of 5 podcasts about the Antarctic Research Centre's ANZICE program. In this episode, we'll turn our attention away from Antarctica and take a look at some earth science being carried out in our own backyard. The deep sea floor surrounding New Zealand is continuously capturing sediments that are eroded off the adjacent landmass. Incorporated into these sediments are the remains of calcareous marine plankton and grains of pollen from land-based plants. The Southern Ocean New Zealand Responses stream of ANZICE is using this sedimentary archive to better understand the environmental conditions that prevailed both on land and in the ocean during peak warm periods in the past. Gavin Dunbar, who leads the research group, is currently extending the study to see what kinds of records may exist in the highly resolved sedimentary sequences beneath the glacial lakes of the central South Island. Welcome to the show, Gavin. Thanks for your time today. Now, in the last episode, we talked to Nancy Bertler about her team's work on how Antarctica may respond to a warmer world. What's the overarching theme of your research group?
Well, I guess we're really looking at two things. Mainly, we're focusing on paleoclimate records that we extract from marine sediment cores. So often they're what we call proxy data for things like ocean temperature and ocean productivity. But in the same cores, if they're close enough to land, we can also get a record of changes in vegetation on the nearby landmass. So in fact, we're trying to do two things. One is to look at changes in the ocean over time, but also from these same marine cores, we're trying to extract records of vegetation change on land at the same time.
So your research group is called Southern Ocean New Zealand Responses. Now, this focus on the New Zealand region, I assume, is for more than just logistical convenience. What is special about New Zealand's position on the globe in terms of the sort of work that you do?
There's really only two land masses in the southern hemisphere at mid-latitudes, so there's New Zealand and there's South America. We know from ice core records that there are two kinds of climate signal, if you like, at higher frequency, so things that change on perhaps decadal to millennial scale. And what happens in the northern hemisphere is not necessarily the same kind of thing that happens in the south. So really we're interested in the southern hemisphere response to changes in global warming. So it doesn't necessarily follow that what happens in New Zealand will be the same as what happens in the northern hemisphere. In that respect, we're very interested in specifically what New Zealand does, but we also know that there are linkages through things like ocean currents, deep ocean currents that Lionel will have mentioned, back to Antarctica. So because we have a research focus in Antarctica, it also made sense to link the two together. And also, as you mentioned, there are some logistical advantages to working on material around New Zealand where we have a lot of background knowledge, and also there are other institutions that can directly contribute to our study. So places like NIWA and GNS Science actually have quite a collection of core material already from this region. So that saves us a lot of time and money in terms of going out and collecting new material.
So once you've collected one of these deep sea sediment cores, what sort of information can you glean from that core, and how do you go about getting that information?
Sediment cores are one of nature's wonderful tape recorders, and what tends to happen in the deep sea, so I'm talking about cores that are collected from water depths of 1,000, 2,000 meters, that the accumulation of very fine material, so either the remains of plankton in the water column or material washed out to sea from rivers, accumulates fairly consistently over many thousands of years. So what we tend to do is we'll get the core back into the laboratory and we cut it open, and then the top of the core basically represents the youngest material. So that's sort of, today or within a few hundreds of years. And then often in the deep sea, accumulation rates, the rate at which material accumulates on the seabed, is on the order of perhaps 1 to 5 or 6 centimeters of material for every 1,000 years that's gone by. So several meters of core can actually represent hundreds of thousands of years in terms of climate history. So the first thing we try to do is to figure out what the age of our material is, because when you open it up, it looks like white sand if it's full of plankton, or there's really nothing that's particularly striking about a lot of these marine cores when you first open them up. So really, in order to get the kind of information we want, we need to dig into them and look at them in great detail with the microscope. So the first thing we try to do is to determine the age, and sometimes this can be done by identifying volcanic eruptions, so little thin layers of ash do stand out in these cores. And there's a very famous one, the Kawakawa Tephra, which is, I think, around 27,000 years old, which we actually find all over New Zealand and way out to 1,000 kilometres out to sea. And that's a very good time marker when you can find that. Otherwise, we use sort of geochemical techniques to look at changes in the chemistry of the plankton over time, and we can relate that back to sort of a global timescale, if you like. So we generate these patterns of geochemical change, and then we relate that back to a global stack or a standard where these changes are, instead of depth, they're actually in terms of time. So by that means we convert the depth in our core to time in the past. Once we have an age model, then really the Pandora's box is open, and there's lots of other measurements we can make. A lot of our work is based on chemical analysis of calcareous plankton. In particular, we look at foraminifera, which are sort of I guess you would call it the rats of the sea. They are very widely distributed, not necessarily at very high latitudes. Around the polar regions they're not so abundant, but certainly around New Zealand there's no shortage of them. So they're very widespread, and they also occur reasonably consistently on the timescales that we're looking at, so it's not like they disappear for 10,000 years and then reappear. So a lot of our analytical work is based on those, but also we look at things like fossil pollen grains, because we can relate pollen back to the parent plant that produced it. And so each kind of thing that we analyze requires a different analytical technique and a different laboratory, and each one of these has its own pitfalls and traps for young players. So we basically start with an age model, and then we go through and look at the kinds of things that we can relate to changes in ocean history. So certain chemical changes are known to be associated with changes in temperature in the ocean. Changes in pollen reflect changes in vegetation on land, and ultimately most of these changes can be related back to climatic change, the ice ages and things like that.
Compared to Nancy's ice core work that's generally focused on the Holocene, so the last 10,000 years, it sounds like your research is concerned with much longer timescales.
I guess one of the themes that we chose specifically is what's called super warm interglacial periods. So over the last several million years, well, in fact, going back much further than that, there are more than one ice age, if you like, and at least over the last 700,000 or 800,000 years, there's been one ice age about every 100,000 years. But in the jargon, we call that a glacial period. Where we're at the moment is what we would call an interglacial or relatively warm period. But if you go back into the past, there have been some periods of time where it's been significantly warmer than today. And so the previous time this occurred was during what we call isotope stage 5e, which is around 125,000 years ago. If you push it back a little bit further, about 1 million years ago was the last time we have clear evidence for a major retreat or collapse, if you like, of the West Antarctic ice sheet. And so the question is just how warm was it when these major climatic events happened? You know, is it 3 degrees or 5 degrees or 10 degrees warmer? So really we put a lot of effort into trying to quantify just how warm those warm periods were, and then look at the consequences of that warming on things like ocean circulation patterns and the stability of the ice sheet. And what we can do is we try and also look at regional differences, like maybe it was much warmer in Antarctica during certain periods of time, but relatively not so warm in New Zealand. So for that reason, we focus on much longer timescales than perhaps Nancy does. The key events for us were about 1 million years ago. About 400,000 years ago was a very long, extended, warm interglacial period, and about 125,000 years ago is the most recent one. There are a few intriguing events in between where the incoming solar radiation values in the southern hemisphere were very high, but in fact the global climate appears to have been quite cool, and so we've got an interest in those as well. But also there is sort of a necessity factor that deep ice cores that go a long way back in time are actually extremely difficult and expensive to recover, and that is sort of the sport of kings, if you like. So there are huge international programs that do that kind of work. But realistically, for an organisation as the size of Victoria, it makes more sense to focus on more recent periods of time which are more accessible with the equipment we have.
You mentioned before foraminifera, so these are zooplankton or animal plankton.
Yep.
Now, in several previous episodes, we've discussed diatoms, marine algae. These are basically different names for the same sort of thing. That is, the other major type of plankton, phytoplankton or plant plankton. Is that right?
Yes, so there are some things that sort of are essentially microscopic plants that photosynthesize in the ocean, and there are other things that are typically slightly bigger that often eat them, zooplankton, which are animal.
And so phytoplankton would essentially be the base of the marine food chain.
Yeah, that's pretty much it. So changes in the production of those kind of things actually can have quite major consequences through the rest of the food chain. If you have a big change in the production rate of plankton, that can flow through to the rest of the food web. And actually, just as a bit of an aside, one of the other things that we're interested in is the role of iron fertilization on plankton growth in the Ross Sea. And the idea there is that windblown dust contains this iron, which, interestingly enough, photosynthetic organisms need as part of their nutrients. So you think things like nitrogen and phosphorus, but they don't need much, but they also need some iron. And in fact, most of the Southern Ocean is very devoid of this micronutrient. But where you get the dust blowing out onto the sea ice, and the dust accumulates, and then the sea ice melts and dumps this material in the Ross Sea, and you get huge blooms of plankton when you just add this one nutrient that's missing. And so we're interested in how that cycle works as well and how it changes over time.
Another of your current projects is looking at the relative roles of temperature and what's known as insolation in the production of these massive algal blooms in the oceans around New Zealand. Insolation refers to the amount of solar radiation that the Earth receives, and plants obviously need sunlight to grow. Could you briefly explain insolation at the timescales that you're dealing with? Because listeners may be thinking that we're talking about changes in incoming sunlight due to atmospheric phenomena or changes in direct solar output or something like that.
The kind of changes in insolation that we focus on the timescales that we're looking at are typically what's called Milankovitch cycles, after the first person to really put forward a coherent theory on how this works. It's not related to changes in the output of the sun, but it is related to changes in the Earth's orbit around the sun. So over tens or hundreds of thousands of years, the Earth's orbit goes from sort of a relatively circular shape to a more elliptical shape. And also the tilt, what we call the obliquity, changes. So the Earth kind of spins a little bit off center. So in fact, we're tilted over at about 23 degrees or something. And also it sort of wobbles around a little bit like a top. All of these changes change the seasonal distribution of insolation. They don't necessarily really change the total amount of it, but they do change its seasonal distribution over time. Typically, these values are calculated what we call top of the atmosphere. So in terms of the insolation that's actually incident on the ground, yes, things like clouds and all that will play a role, but it's actually very hard to try to quantify that effect. So we tend to take a bit of a shortcut and just use the calculated values at the top of the atmosphere. That's not to say that things like cloud cover are not important. They're simply very hard to actually incorporate into the kind of climate change that we're looking at.
So this study is largely based on a sediment core that shows interesting compositional changes during the penultimate interglacial period, so that's over 200,000 years ago. What's the background of this study, and what are some of the preliminary findings?
Our colleagues at NIWA were kind enough to provide us with a sediment core from a little bit northeast of New Zealand that we were going to use primarily for teaching purposes. And when we had a look at it in detail, it had these very marked layers where you could see that the composition of the plankton had changed. And in one particular period of time, which we traced back to what we call isotope stage 7A, there was a very white, chalky layer that when we looked under the microscope was full of what we call coccolithophores, tiny plant plankton. And one of the things we're interested in is we know that the apparent blooms of these things appear to be increasing today off New Zealand, and we were sort of interested, if we wind the clock back through time, what is the evidence that these things are associated with increased warming? So, for example, marine isotope stage 5e is widely regarded as being warmer than today, and is there evidence for a change in the composition or the productivity of these plankton back then? We were a little bit surprised, I guess, when we saw that, in fact, what looked like the greatest increase in productivity had occurred during the stage 7A. So there is more than one thing that influences the growth of calcareous nanoplankton, and it's very early days in this project, so there's lots of things for us to learn and find out before we sort of deliver a definitive result. But it's clear that the temperature itself, at least for some species, is not necessarily the overriding control, and there are other factors. Irradiance, light levels is one of them. And it turns out that during stage 7A, in fact, the southern hemisphere has very high levels of insolation, and we were wondering really whether that actually represented a significant control on the growth of these particular plankton instead of just primarily temperature. And we compared that to a cooler part of the same interglacial period, so what we call stage 7C. And in fact, then the insolation in the southern hemisphere is very low, and in fact, correspondingly, there is a very low level of production of these particular plankton. Now, what's interesting about the 7A is that globally it's not seen as a particularly warm event. So relatively, it's sort of cooler than today, although it's an interglacial period, but it does appear to have enhanced, and in the case of 7A, greatly enhanced levels of insolation arriving in the southern hemisphere. So what we're trying to do is sort of to untangle all these different factors, the light levels, nutrients, because insolation may change the temperature and density structure of the ocean slightly, which may change the availability of nutrients in the surface ocean. All of these things kind of go into the pot, and I guess what we're trying to do is unravel which ones are the most important. But it does appear that maybe temperature by itself is not necessarily a major control, or the major control at least. But as I said, I need to emphasize that it's sort of early days, and our thinking on this will quite likely change over time.
So in your research group, you have these studies of marine microfossils that show you how the ocean has changed over time. And then there's the work that you're just starting in the Central South Island glacial lakes that appear to have these deep and finely layered sediments that are likely to document some form of terrestrial and or atmospheric change since the last ice age. But it's very early days on that front, so I think we'll leave that for another podcast at this stage. But the third major kind of study your team does is to investigate records of land-based plant pollen captured in marine sediment, so you're able to detect changes in the land environment based on evidence found in the sea. To finish, could you elaborate a bit more on this aspect of your team's research?
Again, this is work that's collaborative with NIWA and GNS Science. We've analysed the pollen from a marine core that sits off, I guess the nearest regional centre would be Hokitika, and over the last 210,000 years, the core that we've got has been slowly, quietly accumulating pollen grains that are flushed out by the rivers into the Tasman Sea. And we know that with certain limitations, we can relate the pollen grains back to the vegetation type on land. And one of the great advantages of working on this material in a deep sea core is it's much easier to determine the age of the material going back through time. On land, you can use radiocarbon dating and things like peat swamps, but really that's a technique that's applicable to the last 35,000 years or so. And for older material, it becomes much harder to determine the age. Whereas because we have access to slightly different geochemical techniques in the sea, it's actually much easier for us to determine the age of this pollen material, as long as the time it's taken to get from the land to the sea isn't too long. And so what one of our students, Matt Ryan's, been able to do is to build a very detailed record of changes in the vegetation on land over the last 200-odd thousand years. There are changes that are associated with the ice ages, so you can see that 20-odd thousand years ago there was a big reduction in warm forest, so we lose things like rimu and other podocarps. It's replaced more by grassland and alpine shrubs. And if you go back, say, to our super warm period, isotope stage 5e, the evidence would tend to suggest from the pollen that we're experiencing not only warmer temperatures in the ocean around New Zealand, but perhaps not surprisingly also warmer temperatures on land. One thing that's difficult to do with pollen is to turn changes in vegetation type into what we call a quantitative measure of warming, so we can get a sense that, yeah, it was probably warmer here, but what we actually want to know is, was it 2 degrees or 5 degrees warmer, that kind of thing. So one of the challenges we face now is to try to take Matt's record and actually quantify it in terms of just how much warmer was it. And we can see that by looking at some work that was done about 15 years ago on the eastern side of New Zealand, a similar study was done on a marine core there that actually covers the same period of time, and we can see that the vegetation changes there are quite different. There is still a warm, cold response, but the kinds of vegetation in detail and the changes in the actual timing of those changes appears to be slightly different between the eastern and western side of the Southern Alps. And I guess what we're trying to do now is that one of the other challenges, apart from simply trying to figure out how warm these really warm periods were, was to look at, is there a different response in different parts of New Zealand? Is there a sort of a regional difference related to perhaps how far the vegetation is from the sea, which side of the Alps it's on? We're assuming that rainfall isn't really a limiting factor on the west coast. As we all know, it's pretty wet. But on the east coast, rainfall becomes much more of an issue. So again, what we're trying to do is take our records, our proxy records, if you like, for climate change, and then try to tease out the details which allow us to understand how the climate system in this part of the world works in more detail than we've been able to do it before.
For more journeys to the ice, visit sciblogs.co.nz/journeys to the ice.